So we have started with our industrial pharmacy. We have seen two chapters from physical pharmacy, micromeritics and rheology. So remaining chapter also will take simultaneously. So I'm starting industrial pharmacy and your physical pharmacy. So we'll take it simultaneously. From industrial pharmacy, we have started our pre-formulation studies, right? So pre-formulation studies means what? It is nothing but the evaluation of physicochemical properties of a drug which affect the formulation parameters. So we were started with this. So soon after your research, okay, soon after your identification of a molecule, soon after your isolation of molecule from either natural source or soon after the synthesis of molecule from chemical, okay, chemistry lab. So if it has a promising effect in the animal studies, then you have to start your pre-formulation studies, right? Because ultimately you are going to develop that compound, you are going to conduct trials on that compound, but at the time of release you should be have your work done, you should have all the pre-formulation studies done, then only you can market the product. Otherwise what will happen, you will isolate the molecule, you will optimize the molecule, you will find new molecule, you will if you conducted the clinical trials but still you are not able to develop the formulation. Why? Because you have not done pre-formulation studies. So the research department of medical chemistry pharmacology, they have to discover a drug, they have to discover a drug, and then after discovery they have to make a drug in a different form like salt form or solvate form, hydrate form, etc. Or they have to develop prodrugs because solubility is very important. If drug is not soluble in the water, so solubility affects the bioavailability. Solubility will affect the bioavailability. So bioavailability. That's why suppose you have synthesized one molecule or suppose you have identified a molecule, suppose you have isolated from natural plant, right? You have discovered the new drug, right? Now new drug discovery is a very lengthy process. It will take around 15 to 20 years. For a new drug discovery it will take around at least 15 to 20 years, 15-20 साल लग जाते हैं नया ड्रग डिस्कवर करने के लिए, and it will take around $800 million as an expense. So suppose that you have isolated one compound from natural source, or if you have optimized a molecule, if you have synthesized new molecule, right? Then pre-formulation work start. Now what you have to do, you have to optimize that molecule, you have to study the physicochemical properties, you have to characterize that molecule in terms of physicochemical properties. If you don't know the physical properties, chemical properties, if you don't know the solubility, if you don't know the bulk parameters of the drug, so you will not able to formulate the your formulation effectively. You are not able to convert the drug into formulation, because ultimately the formulation you are going to release in the market, right? So drug discovery, that is identification of lead molecule, then isolation, characterization, then formation of stable molecule, and then salt formation to increase the solubility of compound, to increase the solubility. Most of the time, okay, most of the compounds, they are free organic molecules. Most of the compounds are the free organic molecules. Now they have less solubility in the water, less aqueous solubility. So to increase the aqueous solubility, salt formation. Salt formation, like for example, salt formation means what? So it is nothing but the exchange of proton, exchange of proton from molecule to the corresponding ion to form a salt. So either for example is hydrochloric acid salt, sulfate salt, citric acid salt, fumaric acid salt, like that. या फिर otherwise sodium salt, potassium salt, calcium salt, zinc salt, etc. etc. Then again evaluation and selection of drug. Pre-formulation research department, pre-formulation research department, they will formulate, they will identify literature regarding the that molecule, primarily or primarily data available, data on the stability, key stability data, etc. etc. Then finally you have to develop a formulation request. Now in the pre-formulation what you have to do, different parameter you have to study. Physical characterization like bulk drug properties, solubility profile, stability profile, Now, in the formulation development department, compatibility of the drug with the excipient stability, then dissolution, bioavailability. These studies you have to do in the pre-formulation only. Now, once you have done this study, then you can, okay, then you can file the IND, investigational new drug application, to the FDA. IND, IND means investigational new drug application. Now, okay, you can file the IND, and if after filing, FDA will review that IND. If they finds the data given by the applicant is satisfactory, then they will approve. Okay. Once they will approve IND, then you have to do clinical trials. You have to do clinical trials. Right? After the clinical trials, once clinical trials are over, and if it is promising, then you have to you know you have to do formulation. You have to market the drug. Now, process research, in between that process research will improve the yield or alter, okay. So alternate route. For example, if you have synthesized that molecule and it is costing you a lot of it is expensive to synthesize that drug. So improvement of yield, alternate route for synthesis, production in the bulk. Then once you have developed the formulation, then bulk scale up. So you have to manufacture that formulation on a large scale, right? Then analytical development, okay. This is the role of process and research development, okay. Then analytical development department का role होता है to develop a assay technique, to develop a dissolution technique, to develop a CU technique, continuous informatics technique. Okay. So analytical development, there is a bulk clearance, toxicology potency, formulation assay, and the formulation stability. And lastly, bioavailability studies, toxicology studies. We have seen this in the previous session also. I would like to revise you, and we'll take another further points also. I hope I am audible to you clearly. Why we have to do pre-formulation? Pre-formulation is mainly done for, mainly done for to know the 3 points. Pre-formulation is done for either to know the physicochemical properties of new drug, to know the kinetics and stability of drug, to ensure the compatibility with the commonly used excipient. Because ultimately you are going to formulate that drug in a formulation. Pre-formulation किसके लिए करना है भाई? पहला तो ये आपको फिजिकल केमिकल प्रॉपर्टीज मालूम होना चाहिए वो ड्रग का. दूसरा kinetics क्या है उसका, instability क्या है, to ensure the compatibility with the excipient, so drug, once a drug will, if you find a drug with a promising effect in the animals, which assures you or which will give you warranty to evaluate that drug in humans. So pre-formulation is nothing but it deals with the study of physical chemical properties of drug that could affect the drug's performance, or simply drugs that could affect a drug in terms of therapeutic effect also, and development of dosage form in the patient, for the patients. So understanding this property helps in the formulation design. Formulation design में help करेगा कौन? Pre-formulation. It helps in the formulation design, supports the molecular modification if any required. अगर आपका compound optimized नहीं हुआ है तो you can optimize that. And also pre-formulation will definitely help you to build up the formulation, but also to okay, but also to you can also know the process. You can also know the process through which you can develop formulation and you can market that. Okay, the formulation has impact on the manufacturing, then storage, performance of drug product. Performance means effect. The formulation study provides a way through which drug product can be processed and stored to ensure the quality of that. ओके. तो ये अपने देखा है प्रीलिमिनरी प्राइमरी फाइंडिंग्स एंड मॉलिक्यूलर ऑप्टिमाइजेशन. आफ्टर दैट solid state physical properties of drug, in that compaction properties में molar volume density, refractive index, thermal and electrical conductivity, hygroscopy. Molar volume density will definitely affect the population. अगर बहुत ज़्यादा dense है material, if you want to formulate in the capsule, so obviously you have to use the gelatin which is having not the parameter. gelatin having more bloom strength. Okay? If your material is highly dense, then you have to use gelatin of high bloom strength, because that should withstand with the volume, or that should withstand with the mass of that material to hold. अगर हाई जेल्टिन ब्लूम जेल्टिन यूज नहीं करेंगे, अगर मटेरियल आपका डेंस ज़्यादा है तो वो कैप्सूल जो है ना टूट जाएगा, राइट. सो कॉम्पैक्शन प्रॉपर्टी आल्सो यू हैव टू take into consideration: molar volume, density, refractive index, thermal and electrical conductivity properties, hygroscopicity. You have to take in thermodynamic property like melting point कितना है, इसका melting range क्या है, sublimation temperature, internal energy means internal energy, then enthalpy, heat capacity, entropy, free energy, chemical potential, thermodynamic activity, vapor pressure, solubility. ये सब आपको चेक करना पड़ता है प्री फॉर्मुलेशन. Spectroscopic analytical properties like electronic properties by UV spectroscopy, vibrational by IR spectroscopy, rotational by microwave, and nuclear spin properties by NMR transitions. Kinetic properties like dissolution rate, solid state reaction rate, stability. Surface properties like surface force, surface free energy, surface and interfacial tension, crystallinity of that molecule. Mechanical properties like hardness, tensile strength. और वो mechanical properties नहीं है उसमें. If your material upon a compression, it get deteriorated, then it is useless. You cannot formulate that material into tablet. Hardness, tensile strength, compactability, flow properties, and bending properties of that. Salt formation मतलब क्या, organic substances by abstracting a proton or by donating the proton, by abstracting the proton or by donating proton to form a ionized drug, and then ionized drug is neutralized by using its counter ion. Okay? So ये tomorrow's we'll see in tomorrow's lecture in the detail. Generally organic acids they are more water soluble than unionized drug, so which offers the higher dissolution rate and buyout. So organic acids are more water soluble than unionized drug. For absorption you need an unionized drug. Commonly used anions like enamine, hydrochloric acid, hydrochloride bromide, or chloride, iodide, citrate, malate, phosphate, sulfate वगैरह. Cations मतलब magnesium, calcium, potassium उसका salt बनता है. Pre-formulation studies they are best mainly classified into three types. Solubility analysis, in that you have pKa determination, dissociation constant measurement, pH solubility profile, common ion effect of temperature on solubility, solubilization, different type of solubilization mechanism to increase the solubility, partition coefficient, then dissolution. Bulk drug characterization, in that you have crystallinity, polymorphism, hygroscopicity, micrometric property like particle size characterization, density, porosity, powder flow properties, etc. And in the last, stability analysis, in that solution stability, solid state stability होता है, then drug excipient compatibility. So basically we are going to see pre-formulation chapter under these three headings. तीन main heading रहेगा आपका: solubility studies, bulk characterization, polymorphism and crystallinity, then stability analysis. So पहला होता है bulk characterization, crystallinity and polymorphism. So we have already seen the crystallization chapter. So in that we have seen types of crystal shapes, crystal shape, crystal habit, Meyer's theory of supersaturation. So if you see polymorphism is what? So polymorphism is existence of compound in a different crystalline form, or simply crystallization of compound in a different crystalline form while crystallization. It is a property, or it is a ability of compound to crystallize in a different crystalline form, the polymorphism. Now if you see the compound, any compound under microscope, अगर आपने कोई compound ले लिया माइक्रोस्कोप के नीचे देख लिया, it is either transparent or it is a non transparent. अगर compound माइक्रोस्कोप के नीचे देख लिया आपने, ये दो possibility है. एक है transparent compound है. Transparent compounds they are known as a birefringents. ट्रांसपेरेंट अगर है तो बायफ्रिंजेस बोलेंगे उसको ना, अगर नॉन ट्रांसपेरेंट है तो वो नॉन ट्रांसपेरेंट. Transparent means birefringes. So now it means it will have a refractive index. Right? Now if it is the compound is having one or more refractive index, say for example 2, 3, 4, etc. So if compound has a one or more refractive index, more than one refractive index, it is known as anisotropic. It is known as anisotropic. Now if it is having only one refractive index, known as isotropic. Now, in the more refractive index, you have two. So, if compound is having two refractive index, means uniaxial. If it is having three, means biaxial. Non-transfer जो compound है, it can be described in two terms: internal structure as well as external structure. Internal structure can be divided into two types. External structure is known as a habit. तो इसके ऊपर क्वेश्चन आता है, external structure of compound is nothing but the habit. Habit में भी different shape होते हैं. Internal structure is divided into two types. First one is a crystalline one, second one is the amorphous one. Right? What is crystalline? There is a systematic arrangement of particles in the crystal lattice, and therefore there is a low energy in the system. The system is more stable. तो crystalline form जो होता है वो stable ज़्यादा होता है, उसमें energy कम होता है, energy कम है. जो unstable होता है ना, unstable, इसमें ज़्यादा energy है, तो एनर्जी एक्सपेंड करने का ट्राई करता है, तो उसका सोलिडिटी ज्यादा है. तो अनस्टेबल अगर सिस्टम है तो दैट हैज अ मोर सोलिडिटी, राइट. दैट्स व्हाई क्रिस्टलाइन फॉर्म ऑफ कंपाउंड हैज अ लो सोलिडिटी. एंड अमॉर्फस मींस व्हाट? रैंडम अरेंजमेंट ऑफ पार्टिकल इन द क्रिस्टल लैटिस, हाई एनर्जी एंड देयरफॉर हाई सोलिडिटी. इट हैज अ हायर सोलिडिटी इन द वाटर. So remember, amorphous form of compound has a high solidity, and in between these two there is one more transition state or intermediate compound known as a metastable. So remember, they used to ask you a question like arrange the following form of compound in their increasing order of solidity. Increasing order क्या होगा? Crystalline is less than metastable is less than, sorry, crystalline is more than, sorry, crystalline is less than metastable is less than, no, amorphous. So amorphous form of compound has the highest solidity. Then after that metastable, and सबसे कम solidity होता है crystalline का. You have to remember, okay? Are you getting or not? Are you getting or not? क्या याद रखना है, metastable वाला form होता है, crystalline form होता है. So crystalline form has the lowest solubility. After that, and after that metastable, and after that metastable form, and after that amorphous. So amorphous has the highest solubility. Because in the amorphous there is random arrangement of particle in the crystallized, and therefore system is unstable, and therefore there is a higher solubility, higher energy, right? Now in the crystalline form, okay, now amorphous form they are prepared by this method: rapid precipitation, lyophilization, and rapid cooling of solvent during crystallization. तो ये methods के ऊपर भी एक क्वेश्चन आ चुका है, how you can prepare the amorphous form. Now if solubility is less, you know that crystalline form has a less solubility. And if you want to improve the solubility, obviously you can convert crystalline form into amorphous form. So one of the technique to improve the solubility is nothing but the conversion of crystalline into amorphous, because amorphous is unstable and has higher solubility. Now crystalline form is divided into two types. First one is a single polymorph, or second one is nothing but the molecular adduct. सिंगल पॉलीमॉर्फ मतलब क्या कि इसमें कोई भी वाटर ऑफ क्रिस्टलाइजेशन, सॉरी, सॉल्वेंट ऑफ क्रिस्टलाइजेशन या फिर वाटर ऑफ क्रिस्टलाइजेशन नहीं है. सो इन द सिंगल पॉलीमॉर्फ, ओके. मोलिकुलर एडक्ट मींस इसमें क्या है, ड्यूरिंग क्रिस्टलाइजेशन देयर इज एंट्रैपमेंट ऑफ सॉल्वेंट ड्यूरिंग द क्रिस्टलाइजेशन. यहाँ पे क्रिस्टलाइजेशन के वक्त क्या होता है, इसमें सॉल्वेंट आ जाता है. सॉल्वेंट अगर वाटर है या फिर दूसरा भी हो सकता है, ऑर्गेनिक सॉल्वेंट हो सकता है, वाटर भी हो सकता है. Single polymorph means there is no solvent of crystallization, right? So in that also you have two types: monotropic and anisotropic. Monotropic means all polymorphs are unstable at temperature and pressure, and anisotropic means all polymorphs are isolable. Now in the molecular adduct you have two types. Now molecular adduct means क्या? Molecular adduct means during the crystallization the solvent medium or solvent of crystallization will get entrapped into the molecule. Now ये जो entrapment होता है molecule मतलब crystallization के क्रिस्टल में जो solvent ऐड होता है वो कैसे हो सकता है? एक तो इक्वल प्रोपोर्शन में हो सकता है. Now, whatever solvent which is entrapped during the crystallization, it may be equal. So it may be 1:1. So if the solvent entrapped during crystallization, if and the crystal, if it is 1:1, it is a 1:1. It is known as a stoichiometric molecular adduct. Right? If it is a 1:1, in that you have solvates and hydrate. Solvates मतलब क्या? If crystal consists of a solvent of crystallization, that is known as solvates. And if that solvent is a water, it is known as a hydrate. अगर solvent water है तो उसको hydrate बोलेंगे. तो अगर एक molecule है, if one molecule of water is there in a crystal known as a hemihydrate. Sorry, if half molecule is there in a crystal known as a hemihydrate. If one molecule of water is there in a crystal known as a monohydrate. If two are there, dihydrate, trihydrate, polyhydrate, etc. Tetrahydrate, and like that. Non-stoichiometric. Now, stoichiometric means 1:1 proportion. Non-stoichiometric means okay, there is no proportion 1:1 एक नहीं हो सकता. तो मतलब नॉन स्टोचियोमेट्रिक. स्टोचियोमेट्रिक में तो 1:1 अगर प्रोपोर्शन में ऐड हुआ है वाटर या फिर जो भी सॉल्वेंट है उन्हें स्टोचियोमेट्रिक. नॉन स्टोचियोमेट्रिक मीन्स नो प्रोपोर्शन. यहाँ पे प्रोपोर्शन कम है, नॉट 1:1, 1:3, 4 ऐसे भी हो सकता है. ओके, 1:2 से यहाँ पे लिखता हूँ 6:4 कुछ भी लिखो, not a standard. So in the non-stoichiometric there are three types: cage, layer, channel. Now after addition of this solvent or water crystallization in the molecule non-stoichiometrically, they will develop into different shapes like cage-like shape known as a cage or layer and channels. इसके ऊपर भी क्वेश्चन आ चुके हैं, लास्ट ईयर भी आ चुके हैं. Now we have seen the habit, acicular, angular habit. Okay, so I would not take it again. We have seen the crystal habits. Polymorphism, ability of compound to crystallize more than one crystalline form. To crystallize in a more than one crystalline form. For example, जैसे आपको होता है कोका बटर. कोका बटर है ना, it is used as a base, suppository base in the suppository cocoa butter. Cocoa butter will crystallize in a different form. Will crystallize in a different form depending upon the temperature. जैसे अल्फा फॉर्म होता है उसका, बीटा फॉर्म होता है, अल्फा, बीटा, गामा, डेल्टा, gamma, delta. There are different forms of cocoa butter. Okay, cocoa butter suppository. If you okay, so but out of that most stable form of cocoa butter is nothing but the beta. Beta form is a stable form. If cocoa butter forms a polymorphism and if alpha form is there, alpha form बन गए during the heating. वो heating किया आपने, alpha form बन गए. So alpha form it will melt in your hand only. It will melt at room temperature only. अगर रूम टेम्परेचर के ऊपर ही मेल्ट हो रहा है वो आपके हाथ में, तो फिर सपोजिटरी इंसर्ट कैसे होंगे? दैट्स व्हाई बीटा फॉर्म इज स्टेबल. सो यू हैव टू टेक केयर ड्यूरिंग द melting of cocoa butter base while formulating suppository. You have to use proper temperature. You have to you don't have to expose that cocoa butter to a higher temperature. That's why polymorphism is nothing but the existence or ability of compound to crystallize in a different crystalline form. Now these different crystalline form will have different physicochemical properties. Every crystalline form will have different physicochemical properties, and that's why we have to study it. Ability of compound to crystallize in a one or more crystalline form. Now these different crystalline form will have different physical and chemical properties, and that may affect the formulation. So one, as we have seen, amorphous form has a more solubility, crystalline less. So obviously solubility will affect the bioavailability. Solubility will affect the bioavailability. Chloramphenicol palmitate, for example, it has a three crystalline form, one amorphous form, A, B, C. These are the three crystalline form, one amorphous form. Out of this, B form is more stable. Physicochemical properties they will vary with the internal structure of solid. That example, melting point change हो सकता है, hardness, solubility, optical properties, vapor pressure. They can be changed depending upon the polymorph. That's why you have to study polymorphs. So different methods are used to study the pre-formulation, and for example microscopic method is used. For that you need only 1 milligram sample. Fusion method, hot stage microscopy, particularly the melting properties of compound, उसमें heat of fusion वगैरह. So 1 milligram sample is required. Differential scanning calorimetry, differential thermal analysis 2 to 5 milligram sample. हायर स्पेक्ट्रोस्कोप के लिए लगेगा आपको 2 to 20 milligram sample. X-ray diffraction, so you can study the crystal shape, crystal pattern, for that you need a 500 milligram sample for X-ray. Now crystal purity can be checked by differential scanning calorimetry. You have to remember crystal purity, crystal shape, crystal structure, crystal internal structure you can study by X-ray diffraction. Scanning electron microscopy you need a 2 milligram sample. TGA के लिए 10 milligram sample है. Resolution स्टेबिलिटी के लिए कुछ mg से लेके ग्राम तक सैंपल है. These are the some habits. There is external structure of molecule, platy, equivalent or massive, needle or acicular, bladed, prismatic, tabular, etc. Now, by using microscopy, you can study the external, to study the nature of substance, crystal morphology. By using fusion method, you can study the crystal melting properties of crystal melting point, when softening temperature, etc. TSC and DTA, differential scanning calorimetry, differential thermal analysis. So it is used to study the polymorphism, crystal purity. Crystal purity in the DSC, sharp peak. Sharp peak indicate the pure crystal. दो टाइप के peak आता है. For example ये वाला ना, ये वाला broad peak, ये वाला broad peak. Sharp peak ऐसा, sharp peak. A broad peak. So broad peak indicate impure compound. Sharp peak indicates the pure compound. IR spectroscopy, water content IR से करते हैं आप. Study of crystal also it is done by IR spectroscopy. X-ray diffraction, crystal shape and outer surface of the crystal. Scanning electron microscopy is used for the study the crystal morphology. TGA is to study the crystal. NMR is also used to study the crystal. HPLC is nothing but the stability indicating assay. इसके ऊपर क्वेश्चन आ चुका है एक बार. Dissolution studies it is done by bioavailability. ठीक है, ये ऑलरेडी हो चुका है थोड़ा सा फास्ट. इसके लिए मॉइस्चर कंटेंट इज डेफिनेटली गोइंग टू अफेक्ट द फॉर्मुलेशन, है ना. अगर आपके रॉ मटेरियल में मॉइस्चर है, सपोज योर granules has a more moisture content, so it will not properly flow from hopper to the dry cavity for compression, so it will adhere to the surface of hopper. So obviously moisture content is very important, and moisture content or presence of moisture will also affect the physical stability and chemical stability. So presence of moisture is there in a compound that may cause hydrolysis, the compound may be prone to oxidation. That's why moisture content measurement is important. Okay? Normally जो granules होता है, the granules has only 5% moisture. Moisture content can be determined at different method. Moisture content can be measured by different method. For example, first method होता है आपका simple method, Carl Fisher titration. Carl Fisher titration. Second method होता है tolling distillation method. थर्ड मेथड होता है IR स्पेक्ट्रोस्कोपी ना. तो आपको ये मालूम होना चाहिए कि डिफरेंट मेथड फॉर मेजरमेंट ऑफ मॉइस्चर कंटेंट. टाइट्रीमेट्रिक मेथड होता है, उसको बोलते हैं कार्ल फिशर मेथड. टोलिन डिस्टिलेशन मेथड होता है आपका. ठीक है, बाद में आपका IR स्पेक्ट्रोस्कोपी होता है. Now hygroscopic based upon the moisture content. Yeah. So there are depending upon the nature of compound that will affect the formulation. Hygroscopic compound is there, deliquescent is there. Hygroscopic compound means substance that will absorb the moisture from air and will get there is an increase in the weight of that compound, but the compound will not liquefied. कुछ hygroscopic मतलब क्या, liquid मतलब hygroscopic मतलब if you expose that compound to a humid condition, high humidity में अगर उसको expose करेंगे वो क्या करेगा, मॉइस्चर खींच लेगा अपने अंदर और उसका वेट बढ़ जाएगा, लेकिन इतना नहीं खींचेगा कि पूरा लिक्विड बन जाए. इट इज नोन एज हाइग्रोस्कोपी. Deliquescence means they will absorb sufficient moisture. Deliquescence are the compound which will absorb sufficient moisture from the air and they will become a liquefy, will get dissolved. पूरा लिक्विड बन जाएगा. जैसे मान लीजिए अगर आपका बरसात के दिन है, बरसात के दिनों में क्या होता है, मॉइस्चर ज़्यादा होता है एयर में. अगर उसमें अगर आपने नमक का, आपका जो नमक का पैकेट होता है, you open your salt sachet or packet, and if you keep open in the rainy season, what will happen? That salt will absorb moisture and it will become a liquid known as a deliquescent. There is one more term known as a efflorescent. Efflorescent means a material, and there is one more term, deliquescent, hygroscopic and efflorescent. तीन term होता है. Efflorescent means material which will release the water of crystallization when exposed to humid condition. GPAT 2023 में एक क्वेश्चन आ चुका है. What is meant by efflorescent? The material which will release. तो ये हाइग्रोस्कोपिक वाला, डेलिकेशन वाला, ये मॉइस्चर एब्सॉर्ब कर रहा है ना. हाइग्रोस्कोपिक वाला लिमिटेड मतलब कम अमाउंट में मॉइस्चर एब्सॉर्ब कर रहा है और उसका वेट बढ़ रहा है, लिक्विड नहीं बन रहा. डेलिकेशन वाला इतना मॉइस्चर एब्सॉर्ब करता है कि पूरा लिक्विड बन जाता है. और इफ्लोरेसेंट वाला क्या करता है, ये मॉइस्चर रिलीज करता है when it is exposed to humid condition. So the compound which are exposed to humid condition, they will release the water of crystallization. Okay, water of crystallization known as efflorescent. इसका एग्जांपल है कैफीन, कैफीन साइट्रेट, कैफीन. देन और एग्जाम होता है थियोफाइलिन. और एक एग्जांपल है आपका सोडियम वेलपुरेट, वेलपुरिक एसिड, नेफिडेपिन. दीज आर द efflorescent. हाइग्रोस्कोपिक में बहुत, हाइग्रोस्कोपिक वाला बहुत है. Deliquescent. Change in the moisture level significantly affect the chemical stability and flowability, ultimately compactability of compound or drugs. That's why European Technical Guide, European Pharmacopoeia Technical Guide has classified hygroscopicity based upon the static method. इसके ऊपर GPAT 2022 में क्वेश्चन आ चुका है. क्वेश्चन क्या था वो भी मैं बोलता हूँ. European Technical Guides on the hygroscopicity, they have classified hygroscopicity in the three types. They have classified hygroscopicity in the three types. Four classes based upon the static method. Static method क्या है? You have to take particular weight of powder. Say for example 1 gram of powder आपने ले ली. You have to keep that powder in a stability chamber at 25 degrees Celsius for 24 hours at 80% relative humidity. Now what you have to do? After 24 hours you have to go, you have to withdraw that powder, you have to reweigh. अगर फिर से एक बार वेट करना है आपको, after 24 hours फिर से एक बार वेट करना है. पहले तो क्या करना है, you have to take particular amount of powder 1 gram, that is standard. You have to expose that powder with the condition known as a, you have to expose that powder with the 25 degrees Celsius 80% relative humidity for 24 hours, and then you have to recheck the weight. Now if you have to measure the increase in the weight, you have to measure the increase in the weight. अगर हाइग्रोस्कोपिक है इंक्रीज होगा, नहीं है तो नहीं होगा ना. अगर हाइग्रोस्कोपिक है इंक्रीज हो गया, कितना हुआ? Based upon that, a compound can be called as a slightly hygroscopic. A compound can be called as a slightly hygroscopic if increase in the weight is more than 0.2% weight by weight but less than 2% weight by weight. It is known as slightly hygroscopic compound. If increase in the weight in that compound is a more than 0.02% but less than 15%, known as a hygroscopic. Very hygroscopic means if compound increase in the weight of compound is more than 15%, known as a very hygroscopic. तो इसके ऊपर क्वेश्चन आ चुके हैं. ऐसा नहीं कि हाइग्रोस्कोपिक मॉइस्चर कंटेंट ना, ऐसा नहीं कि हाइग्रोस्कोपिक कुछ एब्सॉर्ब कर लिया, कितना एब्सॉर्ब करता है उसके बेसिस पे उसको क्या बोलेंगे अपन. सो if you take a 1 gram of sample, if you weigh, if you take a 1 gram वेट करो उसका 1 ग्राम एग्जैक्टली कितना आएगा आपका, कुछ तो आएगा उसका प्लस माइनस. You have to take a pre-weighed sample, you have to expose to this condition, and then on the next day you have to take sample and reweigh. Now you have to check the increase in the weight. If increase in the weight of that compound is more than 0.02% but less than 2%, or simply you can say if increase in the weight is less than 2%, if increase in the weight is less than 2% weight by weight, the compound is known as a slightly hygroscopic. Now, if increasing the weight of their compound is a more than 0.2% but less than 15%, or simply if increasing the weight is less than 15%, known as a hygroscopic material. If increasing the weight of that compound is less than 15%, sorry, more than 15%, it is known as a very hygroscopic. तो याद रखना है किसको कब क्या बोलना है ना. आपके मूड में आए तो वैसे नहीं बोल सकते. एक्सपेरिमेंट करके आपको अगर वो कंपाउंड का इंक्रीज इन द वेट अगर 2% से कम है, लेस देन 2%, नोन एज अ स्लाइटली एक्रोस्कोप. 2% से ज़्यादा इंक्रीज द वेट बट 15 से कम है, नोन एज अ हाइग्रोस्कोपिक. अगर 15 से भी ज़्यादा है तो बोलेंगे वेरी हाइग्रोस्कोपिक. If increase in the weight of compound is less than 2%, known as a slightly hygroscopic. If increase in the weight of compound is less than 15%, known as a hygroscopic, and if it is more than 15%, known as a very hygroscopic. इसके ऊपर क्वेश्चन. Now daily question means to become a solution. Daily question means आप solution of water to become a solution. Following are the different methods are used to determine the water content. सो मेथड्स बोला है मैंने कि आपको पहले मेथड है आपका कर्ल फिशर टाइट्रेशन, KF टाइट्रेशन, टू डिटरमाइन द मॉइस्चर कंटेंट इन द सैंपल, KF टाइट्रेशन. इसके रिएजेंट भी होता है अपन देखेंगे. देन उसके बाद में होता है आपका टोलिन डिस्टिलेशन, टोलिन डिस्टिलेशन मेथड. उसके बाद में होता है आपका IR स्पेक्ट्रोस्कोपी, IR स्पेक्ट्रोस्कोपी. There are different methods for measurement of water content in the molecule. KF reagent, toluene distillation method, IR spectroscopy. याद रखना ये methods. Are you getting my voice properly? Hello, am I audible? Audible आएगा? Are you getting or not? समझ रहा है आपको कि नहीं? Following methods are used to determine the moisture content. TGA thermogravmetric method is used. Then gas chromatography is also used. Then gravimetric method is also used. Then KF reagent is used. तो इसमें टोलिन डिस्टिलेशन मेथड एक ऐड करो, टोलिन डिस्टिलेशन एंड IR स्पेक्ट्रोस्कोपी. थर्मोग्रामेट्रिक एनालिसिस, गैस क्रोमेटोग्राफी, ग्राविमेट्रिक, कर्ल फिशर रिएजेंट, टोलिन डिस्टिलेशन मेथड एंड IR स्पेक्ट्रोस्कोपी. नाउ वेंटॉप प्लॉट, इसके ऊपर नाइपर में क्वेश्चन था, व्हाट इज वेंटॉप प्लॉट. इफ यू प्लॉट ए टेम्परेचर वर्सेस मोलर सॉल्विटी, temperature on x-axis, molar solubility on a y-axis. So this plot is known as a Ventop plot. This plot is known as a Ventop. It indicates the solubility pattern of your polymorphs. Different polymorphs will have different solubility at a different temperature. एक polymorph ये temperature पे ज़्यादा soluble है, like this. Molar solubility is nothing but the temperature at which solubility and vapor pressure of polymorph is same. Molar solubility किसको बोलेंगे? It is nothing but the temperature at which the vapor pressure and solubility of polymer is same. Solubility और vapor pressure इसका same है ऐसा temperature पे, इसको बोलेंगे molar solubility. Now bridging solvent is a solvent used to convert one polymer to another polymer. इसके ऊपर क्वेश्चन आ चुका है. Bridging solvent is a solvent used to convert one polymer to another polymer. Fine particle characterization, इसमें अपन ने देखा है ऑलरेडी माइक्रोमेट्रिक चैप्टर में बहुत सारी चीज. bulk flow, formulation, homogeneity, and surface area, control process like dissolution, chemical reactivity, they are affected by this. bulk flow जो होता है, formulation of homogeneity, homogeneous formulation होना चाहिए. surface area, control process like dissolution, chemical reactivity is definitely affected by size, shape, and morphology of compound or powder. Method used to measure the particle size and particle method employed for particle characterization are nothing but the sieve analysis, sieving method, light microscopy, culture counter method. इसके बारे में डिटेल में आपने देखा. स्ट्रीम काउंटिंग डिवाइस होते हैं, स्ट्रीम काउंटिंग डिवाइस भी है. बेड एडसॉर्प्शन जो मेथड होता है, मेजरमेंट ऑफ particle surface area is done by adsorption method. Nitrogen is used to allow to adsorb on a powder to form a monolayer. That adsorption isotherms are given by this equation. Brunauer, Emmett, and Taylor. Brunauer, Emmett, Taylor. Weight equation. So these are the equation is the basis of adsorption method in which liquid nitrogen at 190 degrees monolayer is allowed to form a layer on the surface of particle. Monolayer, not bilayer. Now one nitrogen atom is equal to 16 angstrom in यहाँ पे angstrom 16 angstrom in. तो ये नाइपर के लिए क्वेश्चन आता है. Recently there was one question. There is one apparatus which is used to measure the surface area of particle by adsorption method, known as a quantasol. Quantasol. इसके ऊपर क्वेश्चन आ चुका है. Quantasol is the apparatus which is used to measure the surface area of powder by adsorption method. याद रख. Scanning electron microscopy for surface morphology and texture. है ना, इससे सरफेस मॉर्फोलॉजी, सरफेस कैसा है उसका, टेक्सचर कैसा है कंपाउंड का, दैट इज स्टडीड बाय स्कैनिंग इलेक्ट्रॉन माइक्रोस्कोप. नाउ इफ कंपाउंड इज नॉट हैविंग कंडक्टिविटी, कंपाउंड इज नॉट कंडक्ट, ओके, कंपाउंड में कंडक्टिव नहीं है, सो कंपाउंड इज अ गोल्ड प्लेटेड, compound is a gold plated to make a particles superconducting in the SEM, scanning electron microscope. बल्क डेंसिटी मतलब क्या है, सिंपल मास अपॉन वॉल्यूम. तो मालूम है. Now it is done by passing a sieve through a 40 number. Sorry, bulk density is calculated. Bulk density is calculated of the powder. First you have to pass that powder. First you have to pass that powder through 40 number sieve. You have to pass the powder through 40 number sieve. Then you have to measure the density, mass upon volume. True density is nothing but the bulk density minus by volume. Tap density is nothing but the determined by pouring the known quantity of powder in a graduated cylinder and tapping not less than 1000 times. That is a tap density. इसमें क्या है कि वाइड वॉल्यूम जो है ना, इंटर पार्टिकुलर वॉल्यूम, वो टैपिंग की वजह से माइनस हो जाता है. फ्लो प्रॉपर्टीज दे आर डिटरमाइंड बाय कंप्रेसिबिलिटी इंडेक्स, एंगल ऑफ प्रपोज. फ्री फ्लोइंग पाउडर, पाउडर में भी इधर फ्री फ्लोइंग और नॉन फ्री फ्लोइंग. नॉन फ्री फ्लोइंग मतलब क्या है, कोहेसिव पाउडर होता है. Compressibility or Cars index, or also known as a simply Cars consolidation index. So it is one of the major for flow properties of powder. Percent compressibility is equal to rho t divided by rho 0 divided by rho t minus rho 0. ये क्या है, डेंसिटी. रो टी मीन्स डेंसिटी, टैप डेंसिटी माइनस रो 0 मतलब बल्क डेंसिटी. So tap density minus bulk density divided by tap density into 100 is a Cars index. One of the major part of Hausner's ratio, another measure. So it is nothing but the tap bulk density divided by pore bulk density. मतलब simply tap density divided by bulk density. Simply tap density by bulk density. Hausner's ratio will vary from 1.2 to 1.6. So 1.2 होता है इसके free flowing powder के लिए, 1.6 मतलब cohesive powder. So we have seen already Cars index of powder is between 5 to 12. The powder is free excellent flowing. If it is between 12 to 16, good flow. 18 to 21, possible. If Cars index of powder is between 23 to 35, poor flow is there. If Cars index is between 33 to 38, poor, very poor flow, more than 40, very poor flow or cohesive flow. Okay? Hausner's ratio. If Hausner's ratio of powder is between 1 to 1.11, excellent flowing powder is there. If it is between 1.12 to 1.18, good flowing powder. एयर कब होता है पाउडर फ्लो, इफ का हाउसनेस रेशियो इज बिटवीन 1.19 टू 1.2. ये वैल्यूज इतना इजी नहीं है याद रखना, बट अगर आपने थोड़ा सा 2-3 बार पढ़ लिया तो याद रह जाएगा, इतना नहीं लगता है. हाउसनेस रेशियो 3 वैल्यू है मेरे पास, वो भी याद रखा तो भी चलेगा. इफ इट इज 1.25 टू 1.34, पॉसिबल फ्लो इज देयर. इफ हाउसनेस रेशियो ऑफ पाउडर इज बिटवीन 1.34 टू 1.44, 1.45, poor flow is there. If it is between 1.46 to 1.49, poor flow, very poor flow. More than 1.6, very poor flow. यहाँ भी इजीली लग सकते हो आपको. देखो पहली बार में क्या याद रखना है, लेस देन 11, लेस देन 1.11, एक्सलेंट. 1.112 ये एक्सलेंट है. इसके बाद में 1.12 आएगा, 1 के बाद में क्या आता है 2. तो 1.11 टू 1.18, 18 के बाद में 19 आएगा. 1.119 टू 1.25 यहाँ पे ना 1.11, 1.18, 1.25 पेयर फ्लो, 1.25 के बाद में था 1.26, 1.34 पॉसिबल, 3 4 के बाद में 3 आएगा, 11 ये 20 तक आएगा, 11 से 18, 11 18 25 है ना, ऐसा याद रखो, याद रहेगा। Angle of reverse means what? It is again a measure of your flow properties of water, not a precise measurement, but it is commonly used to describe the flow of water. What is angle of reverse? Tan theta is equal to 2h upon d. Here dh is nothing but the height of the pile, d is the diameter of the pot. Yeah, 2h upon d. इसको और भी एक फॉर्मूला है, simply h upon r. tan tan theta is equal to h upon r. h upon, क्योंकि r क्या है, r यहाँ पे रेडियस है, तो डायमीटर क्या होता है, डायमीटर डिवाइडेड बाय 2 नथिंग बट द रेडियस, तो वहाँ पे 2h upon d ये फॉर्मूला भी करेक्ट है और h upon r ये फॉर्मूला भी करेक्ट है. So static angle of repose is measured by fixed Fresnel method or fixed cone method. Dynamic or kinetic angle it is measured by rotating cylinder method or tilting box method. Tilting box. Now, angle of reverse of water is like if it is 25 to 30. Excellent flow. Yeah. If angle of reverse of water is between 31 to 35, good flow. If it is between 36 to 40, fair flow. 41 to 45, possible. Angle of reverse of water is between 46 to 55, poor flow. And 56 to 65, very very poor, very poor flow. More than 66, very poor. हर एक बुक में अलग अलग दिया है थोड़ा सा, बट एक एक वैल्यू है मोस्ट कॉमनली होता है, राइट. वी आर सीइंग दिस, okay, we have seen this. Now particle size determination method, we have also seen this in the micrometrics chapter. Sieving method, you can measure the particle size in the range of 20 to 20 micron to 1.25 lakhs, sorry, micron. Sieving method, you can measure the particle size in this range to 120 micron. If it is a electrode each sieve. Sieving, if perforated plates 1000 to, then 1 lakh 25,000 micron. Optical microscopy 1.5 to 150 micron. Electron microscopy 0.001 to 5 micron. Gravity sedimentation method by gravity 1 to 50 micron. Centrifugal sedimentation 0.01 to 5 micron. Filter water method 1 to 200. Light scattering 1 to 1000 micron. Compressibility index of few compounds. Cellutab is Cellutab, it is a trade name of cellulose derivative. It is a 11% compressibility. 11% compressibility. So it is having excellent flowability. So in compress is again trade name. So 15% compressibility है, excellent flowing है. Star RX, Star X 1500. These are directly compressible stars. Lactose लैक्टोज मोनोहाइड्रेट 19% होता है इनका, इसका कंप्रेसिबिलिटी. सो फेयर पॉसिबल फ्लो. मेज स्टार्च एंड डीसीपी ड्राई कैल्शियम फॉस्फेट कोर्स इट हैज 27% कंप्रेसिबिलिटी. सो पुअर फ्लो. 31% कंप्रेसिबिलिटी होता है मैग्नीशियम स्टील का, पुअर फ्लोइंग है. टाइटेनियम डाइऑक्साइड 34 होता है, वेरी पुअर फ्लो है इसका. डाई कैल्शियम फॉस्फेट फाइन होना, इसका वेरी वेरी पुअर. टाल्क सबसे ज्यादा पुअर फ्लो. तो टाल्क का परसेंट कंप्रेसिबिलिटी एक बार पूछे. कार्स इंडेक्स ऑफ टाल्क 49. Solubility analysis, what is meant by solubility? It is nothing but the temperature, sorry, it is nothing but the concentration of compound. Temperature it is nothing but the temperature at which solution is equilibrium with the solid particles. This is solubility. So focus on the drug solvent system that occurs. Solubility analysis mostly focusing on the solvent system, solvent system that involves that affect the bioavailability of your drug. The analysis involves the solubility analysis is done by using HPLC. HPLC मतलब high performance liquid chromatography, UV, GC, fluorescence spectroscopy. तो इसमें क्या क्या है, determination of pKa, measurement of pKa dissociation constant, then pH solubility profile, then temperature dependence, temperature अगर increase होगा तो कितना solubility increase होगा, dissolution, stability of product, then solubilization mechanism. So under the solubility analysis you will study these things. First one is a determination of pKa dissociation constant. What is dissociation constant? It is the dissociation constant of the drug. Okay, so percentage ionization is nothing but the a compound will be dissociated into ions, you know, let's say H plus OH minus, two ion H2O, so H plus, so equilibrium between two ions, if two ions are ionized, equilibrium, in equilibrium, one is the ionization. Okay, the pH pKa is nothing but the dissociation constant at which there is an equilibrium ionization of both component of the material. मटेरियल में दो कंपोनेंट जैसे आपका सोडियम हाइड्रोक्साइड NaOH, तो pKa मतलब क्या, the pH at which there is a dissociation, there is a ionization of both functional group, the molecule, right? Ionized drug is a lipid, unionized drug is lipid soluble, therefore easily permeated through membrane. Ionized drug is not lipid soluble, therefore it is less permeated through lipid membrane. But ionized drug is a more water soluble. This is given by one equation known as Henderson-Lewis equation. इसके ऊपर भी एक दो बार क्वेश्चन आ चुका है कि pKa or dissociation constant is measured by following equation. It is determined by Henderson-Hasselbalch equation. So for a basic compound, for acidic compound, it is a different. So pH is equal to pKa plus ionized divided by unionized. That is for a basic compound. For acidic compound, pH is equal to pKa unionized fraction of drug divided by ionized fraction of drug. इसको अपन उल्टा भी लिख सकते हैं, है ना, pH minus pKa is equal to pH minus pKa. अगर ये pKa इधर गया माइनस होता है, pH minus pKa is equal to, है ना, ionized ionized. For basic ionized ऊपर रहता है unionized, है ना, for acidic unionized ऊपर है ionized नीचे. Percentage ionization is equal to 10 raised to pH minus pKa divided by 1 plus 10 raised to pH minus pKa. Now percentage ionization is measured by different methods. Spectroscopic method होता है, potentiometric method होता है, titrimetric. So important है, it is very important methods for measurement of pKa. So spectroscopy में क्या है कि the pK and the change in the absorption of maxima, change in the absorption maxima as a function of pH, intense pK, spectral shift is detected and it gives you idea about the pKa of the molecule. And UV spectroscopy is a preferred method for measurement of pKa. Another method is there known as a titrimetric method, potentiometric method, and in the potentiometry, half wave potential is nothing but the pKa. In the titrimetry, जो half pH होता है, half pH of neutralization. In the titrimetry, half pH of neutralization is nothing but the corresponding to pKa. In the potentiometry, half wave potential is corresponding to the pKa. Now, effect of temperature on the solubility. एक temperature increase होगा तो solubility क्या होगा? So if there is a 10-degree change in the temperature, there is a 5-fold times change in the solubility, है ना. अगर किसी कंपाउंड का solubility अभी मान लीजिए कि 5 ग्राम पर 100 ml है, 5 ग्राम पर 100 ml कंपाउंड का solubility है, और 10 डिग्री सेल्सियस टेम्परेचर आपने उसका अप्लाई किया तो 5 टाइम्स solubility उसका बढ़ जाएगा. Effect of temperature on solubility, इसके ऊपर नाइपर में क्वेश्चन है. If there is a 10-degree change in the temperature, it will increase the 5 times solubility. Dissociation constant is measured by Anderson-Hasselbalch equation and is used to estimate the percentage ionized or unionized drug. Ionized drug are lipid soluble and they are absorbed faster. Unionized drug are lipid soluble and they are absorbed faster. Ionized drug are water soluble drugs. Now इसके ऊपर क्वेश्चन आ चुका है. Now for weekly acidic drug कौन होता है जिसका pK less than 8 होता है. For weekly acidic drug where of which pK is less than 8, such drugs for example nitrile ethosuximide, they are the weekly basic drug. pH is having the pK is having less than 5, such as diazepam, theophylline. They will remain unionized throughout the GIT and completely absorbed. Now weekly acidic drug, pH sorry इनका pK कितना है 3 to 5. So weekly acidic drug having pH 3 to 5. So they are the weak acidic drug. Okay, they are the weak acidic drug and they will absorb from the stomach only, because in the stomach they will remain unionized and most of the weak acids they are absorbed from the stomach only. Aspirin, ibuprofen, they are ionized in a gastric pH, absorbed completely. But at the neutral or basic environment, they will get ionized and they are poorly absorbed. For weekly basic drug having pH in the range of 5 to weekly basic drug having pK in the range of 5 to 8. For example, morphine, chloroquine, they remain unionized in the basic pH and therefore they are absorbed from the intestine. When acidic pH is there, they will get ionized and no absorption. Strong acidic drug is for example bromelain sodium, and strong basic drug for example erythromycin, papaverine. They are ionized in the both stomach and intestine and they are very poorly absorbed. And जो strong acidic drug है या फिर जो strong basic drug है, okay, it is ionized in the GIT and they are poorly absorbed. Methods for measurement of pK, it is a potentiometric method, potentiometric tradition. pK in the range of 3 to 10 that can be measured by potentiometry. तो ये एक in this method there is a lot of variations, lot of factors which affect. ठीक है, concentration होना चाहिए range, particular concentration में ही आप determine कर सकते हो pK. For that, so new this Yeshuda and Sedolowski, so they invented the modified this method. ड्रॉबैक क्या होता है उसमें कॉन्सेंट्रेशन होता है उसका प्रेसिपिटेशन होता है. दैट्स व्हाई ये नया प्लॉट यशोदा सेडोरस्की, ये कल भी अपन में थोड़ा डिटेल में बोलूंगा उसका. दे दिस प्लॉट बाय एक्सट्रापोलेटिंग ओके द स्पेसिफिक पोटेंशियल वो मेजर किया जाता है इसमें. और ये अपन इसके बारे में कल मैं आपको डिटेल में बोलता हूँ. ये थोड़ा सा नया पार्ट है. नाउ मोस्ट कॉमनली यूज्ड मेथड most commonly used solvents for potentiometric titration for measurement of pK, that is alcohol, DMS or dimethyl sulfide, DMF and dimethyl formide, THF. Then they are the solvent. Methyl alcohol is the most common. Trimetric method में weak acidic drug versus standard base half 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 neutralization point जो होता है that is equal to the pK. इसके ऊपर क्वेश्चन आ चुका है. In the trimetric method, weak acidic drug versus standard base they are titrated and the pH of half neutralization. pH of half neutralization is nothing but the pK. In the UV spectroscopy, spectral shift give you idea about the pK. HPLC, capillary electroporation, these are the other methods. Now solubility terms I would like to explain यहाँ पे. Solubility terms are very important. Question इसके ऊपर भी आता है. Solubility terms can be okay. Solubility of drug in the particular solvent or water is expressed in the form of number of parts. Okay. So parts of solvent required to solubilize or to dissolve the one part of solute. For example, जैसे मान लीजिए solvent तो ml में होता है ना, और जो कंपाउंड है वो मिलीग्राम में होता है. So parts of solvent required to solubilize to dissolve one part of solute. एक मिलीग्राम सब्सटेंस के लिए डिसॉल्व करने के लिए कितना सोलवेंट लगता है ना. तो मतलब 1 ml लगता है या फिर 2 ml लगता है या फिर 3 ml लगता है या 10 ml 20 ml 30 ml 100 ml लगता है. तो वैसा एक पार्ट बोले तुम 1 मिलीग्राम भी ले सकते हो, आप 1 ग्राम भी ले सकते हो. सो parts of solvent required to solubilize the one part of solute. Now if less than one part of solvent is required to solubilize one part of solute known as a the solid is known as a very soluble. अगर 1 मिलीग्राम सब्सटेंस डिसॉल्व करने के लिए 1 ml से भी कम सॉलिड लग रहा है तो वो सब्सटेंस जो है वो क्या है, वेरी सोल्यूबल है. If less than one part of solvent is required to solubilize a one part of solute known as a very soluble. Are you getting? Am I audible to you? Am I audible? Yes. Okay. So if less than one part of solute is required to solubilize one part of solute, known as a very soluble. If 1 to 10 parts are required, it is known as a freely soluble. If 10 to 30 parts of solvent are required to solubilize only one part of drug, then it is known as a soluble. And if 10 to if 30 to 100 parts of solvents are required to solubilize only one part of solute, known as a sparingly soluble. देखो ना आप, अगर है ना एक पार्ट से कम लग रहा है तो वो फ्रीली सोल्यूबल है. एक से लेके 10 पार्ट सॉल्वेंट लग रहा है, मान लीजिए टू डिजोल्व 1 मिलीग्राम सॉल्वेंट यू रिक्वायर्ड 6 ml ऑफ सॉल्वेंट, इट इज अ फ्रीली सोल्यूबल. ठीक है, to dissolve 1 milligram solute you require 25 ml solvent, then it's soluble. Okay, then what to dissolve 1 milligram of solvent you need a 50 ml solvent, then it is a sparingly soluble. Likewise that. So if less than one part of solvent is required to solubilize one part of solute, really soluble or very soluble, very soluble. And 1 to 10 part really soluble, 10 to 13, 10 to 30 soluble, 30 to 100 sparingly soluble. Then now 100 to 1000 slightly soluble. Okay, 1000 to 10,000 very slightly soluble. More than 10,000 practically insoluble. Now these solubility terms are as per the US United States Pharmacopoeia. As per the USP, solidity terms are very important. इसके ऊपर बहुत बार क्वेश्चन आता है ना. क्वेश्चन अलग अलग टाइप से आता है आपका. A compound is considered as a very soluble. If a compound is considered as a very soluble, देखो क्वेश्चन कैसे आएगा ऐसा. A compound is considered as a very soluble if ऑप्शन में आएगा less than one part of solute is required to solubilize one part of solute. 10 to 30 part of solvent are required to solubilize one part of solute. If there is option A, 30 to 100 parts of solvent is required to solubilize one part of solute. और एक option है you know like this 100 to 1000 parts of solvent is required to solubilize one part of solute. तो याद रखना है very soluble का मतलब होता है less than one part of solute is required to solubilize one part of solute. very soluble के ऊपर क्वेश्चन ऑलरेडी आ चुका है, ये soluble के ऊपर क्वेश्चन आ चुका है, इसके ऊपर भी क्वेश्चन आ चुका है. Solubilization means what? Solubilization means most of the drugs or most of the compounds they are not soluble in the water, not soluble in the water. To formulate and to increase the bioavailability, you need to increase the solubility of that compound. Now solubility, how to increase the solubility? There are different methods to enhance the solubility, like using co-solvent, then formation of that compound into ester, amide, then again developing the prodrugs. You can enhance the solubility of compound by converting into different salt, by converting into prodrug, by converting into amide, by enhancing the solubility by using co-solvent. Now that is known as solubilization, to increase the solubility of compound in the solution using co-solvent like ethanol, propylene glycol and glycerin. So इसके ऊपर क्वेश्चन आ चुका है. The most commonly used co-solvent, the most commonly used co-solvent, उसका आंसर है आपका, है ना, अब उसका आंसर आया था आपका ग्लिसरीन, प्रोपिलीन ग्लाइकोल, इथेनॉल. So what is that? Solubility of poorly soluble compound can be increased. The solubility of poorly soluble non-electrolyte, non-electrolyte, the solubility of poorly soluble non-electrolyte can be increased by mixing them with the solvent in which compound is already soluble. So it is known as a co-solvency. The phenomenon of increasing the solubility of weak non-electrolyte, weak electrolyte and electrolyte, non-electrolyte. Okay, so phenomenon of increasing the solubility of poorly soluble non-electrolyte by addition of water miscible solvent in which the gas good solubility is known as a co-solvency. इसके ऊपर भी क्वेश्चन में जीपैट में क्वेश्चन आ चुका है. इवन ये डेफिनेशन के ऊपर भी क्वेश्चन आ चुका है, co-solvency. What is co-solvency? It is the phenomenon of enhancing the solubility of poorly soluble non-electrolyte by using water-soluble solvent in which the drug is having good solubility. The phenomenon is known as a co-solvency. This was question in GPAT 2021. Again, most commonly used co-solvent. इसके ऊपर भी क्वेश्चन आया, ग्लिसरीन, प्रोपिलीन ग्लाइकोल एंड इथेनॉल. सॉल्वेंट स्ट्रेंथ मतलब डायरेक्टरी कांस्टेंट ऑफ सॉल्वेंट. Now, glycerol has a solvent strength of 0.5, ethylene glycol 1, ethanol 1.5, DMF and DMA. Solvent strength is nothing but the 4. They are highly polar. DMF मतलब dimethyl formamide and DMA मतलब dimethyl acetamide. So they are having highly polar solvent for the solvent strength. Solubilization co-solvency मतलब क्या होता है, ऑलरेडी बोला है. Solubilization of poorly soluble non-electrolyte, it can be improved by mixing with a stable co-solvent like ethylene glycol, propylene and glycerin. Ethanol. Mechanism by which co-solvent increases the solubility. कैसे increase करते हैं co-solvent, increase करते हैं solubility. Insolubility मतलब क्या है? Why compound is insoluble in the water? Because it will form a hydrophobic bonds. Number of hydrophobic bonds, if it is more, so that compound is poorly soluble in the water. You have to break that bond. So what co-solvent will do? So co-solvent causes the breakdown of that bonds, right? By disturbing the hydrophobic interaction with the water. So what co-solvent do? They will break down the hydrophobic bonds, and that's why solubility is increased. The efficiency of co-solvent for increasing the solubility depends on the chemical structure of drug. More will be the non-polar salt molecule, more will be the solubilization. जितना ज़्यादा हाइड्रोफोबिक मॉलिक्यूल होगा उतना ज़्यादा उसका सोलिबिलिटी बढ़ जाएगा बाय यूजिंग कोसोल. एग्जांपल है हाइड्रोकोर्टिजोन. मोस्टली इट इज एप्लीकेबल फॉर स्टेरॉइडल ड्रग्स एंड प्रोस्टाग्लैंडिन एनालॉग. तो जो स्टेरॉइडल ड्रग है उनका सोलिबिलिटी कम होता है. हाइड्रोकोर्टिजोन एंड हाइड्रोकोर्टिजोन 21 हेप्टेनोइड. तो दो ड्रग है. हाइड्रोकोर्टिजोन एक सिंपल ड्रग, पोर वाटर सोलिबिलिटी, उसका कन्वर्ट किया हाइड्रोकोर्टिजोन 21 हेप्टेनोइड. aqueous solubility is increased. इन दोनों को पे solubility increase किया जाता है by using propylene glycol as a co-solvent. Hydrocortisone 21 heptanoid solubility can be enhanced by using glycol and propylene glycol. Effect of temperature on the solubility. Delta H, that is delta H means the heat of solution. Heat of solution क्या है? Whatever heat is absorbed or released when you try to dissolve a particular substance in the solution. Either it may be exothermic or it may be endothermic. Most of the solubility is endothermic. Most of the solubility is endothermic. Heat absorb होगा. क्यों होता है heat absorb? That's why by increasing the heat you can increase the solubility. Heat released or absorbed when one mole of solute is dissolved in the large quantity of solvent. Mostly process endothermic. Delta H is a positive and solubility is, that's why solubility is increased by increasing the temperature. Solute like lithium chloride and other essential salt. When they will try to dissolve in the water, they will heat the solution, that is known as exothermic, and delta H is negative. In this case, temperature will decrease the solubility. Delta H जो है is obtained from solubility values of standard saturated solution. The control temperature range में determine करना पड़ता है. Okay. So remaining part we'll see tomorrow. थोड़ा सा और भी notes extra करके कल में थोड़ा सा और भी explain. Tomorrow will be the last lecture for this.